So in the previous class, what we saw how exactly we can convert an open chain structure, okay, for example, of open chain structure or a straight chain structure for glucose into a ring or a cyclic structure, okay. And while doing so, what we saw that is always an extra, let me get a pen first. While doing so, when a glucose molecule changes from its straight chain structure to the cyclic structure, what happens? There is an extra stereo center created at the carbon number 1, and we call that carbon as the anomeric carbon. Right? We discussed that in the last class. Okay, and then we discussed that once this cyclization will take place, and we also discussed that cyclization in terms of glucose takes place between the carbon number 1, remember that, and the OH present on the carbon number 5. So you have to know this. So like this is the carbon number 1, because here we have an aldehyde, and the carbon number 5. Okay, and other than that, we discussed that what happens after that when we produce when after the cyclization, we end up producing two different types of the isomers. One is the beta D-glucose, another one is the alpha D-glucose. And glucose produces the glucopyranose, and we discussed that pyranose is a 6-membered ring. Remember that glucose also produces furanose ring, the 5-membered ring, but like this is like a more stable form of the glucose. Like glucose loves to be in a pyranose ring than in the furanose ring. Okay. And these two isomers which are produced, one is called as an alpha D glucose, another one is called as a beta D glucose. So we call them as the anomers. So what are anomers? Anomers are stereoisomers that differ in configuration only at their anomeric carbon. So here I have OH at the top in the beta. In the alpha, I have OH at the lower side. Remember how to memorize that? Lower side is like ants always crawl on the ground. So that's how you remember the alpha. The OH is down. And birds are always in the sky, so OH is at the top, right? So that's the beta. Beta is bird, absolutely, right? So the anomeric carbon of the aldose is C1 always. Can anyone tell me why it is C2 in the ketose? The anomeric carbon in the ketose, why is it C2? Why not C1? And if a sugar is a ketose sugar? Let me see if you have understood the basic thing or if you have revised the previous thing. Double bond, okay. You're very close to the answer, Yara. In the ketose sugars, where do we have the carbonyl group? Ketone. So remember, the intramolecular cyclization takes place between the carbonyl and the OH. So ketone has carbonyl at the ketose sugars have carbonyl here. They don't have it at carbon number 1. So you should know how to identify an aldose and the ketose. For example, if you see here fructose is a ketose. Let me show you. If I give you, look at this. If I give you this structure, you should know that this is fructose because it has carbonyl group at the carbon number 2. So that is why for the keto sugars, which have they have the carbonyl group at the C2 position. So for them, the anomeric carbon is not C1, it's the C2. Remember this. Okay. For aldoses, for all the aldoses, it will be always C1. So for aldoses, it will be C1. For ketoses, it will be C2 because ketoses have carbonyl group at the position number 2. So this is like a recapitulation what we did previously. Okay, so I move on. What we have to do today is, okay, so these are the two anomers, alpha and beta, and moving on further. Okay, how many stereocenters are there? Mohammed says, could we say the hemiacyl carbon is the, you can say that, but we have a different name for it. It's called an anomeric carbon, Mohammed. Call it just the anomeric. Forget about, the whole ring is called an hemiacetal. Okay. So what gives it special? Why is it special? Because an extra stereocenter is created here. Muhammad. How many stereocenters? We have only one stereocenter. Who said that one? No, 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 my God. Two. Oh my God. Very good. That means you have not revised. Two stereocenters? Okay. This is your homework. Find out how many stereocenters are present here. This is so very clear. By just looking at the molecule, I can say how many three. Okay, people are increasing. The numbers are increasing. The numbers are increasing. Wonderful. 5, 4. Finally, I have 5 someone. 3 or 4. You have to be sure. Work out and tell me at the end of the class. Okay. So this is the same thing what I explained. What is the beta and the alpha? Okay. Okay, now that was about the glucose, and then now we'll quickly discuss about the monosaccharides, which are either aldopentoses. When I say aldopentoses, that means they have an aldehyde and the carbon 5. Ketohexoses, they have a ketone group and they have 6 carbons. Say, whenever you have a ketohexose or an aldopentose, they usually form furanose. Remember the 5-membered ring? This one. Glucose forms this one, but the aldopentoses as well as the ketohexoses, they form the furanose ring. Mechanism is exactly same what we discussed, right? It's absolutely same, right? So this is the anomeric carbon here over here, but this is the furanose ring, the 5-membered ring. This is the 5-membered ring. So how do I know this is alpha? I have OH on the opposite side. How do I know this is beta? I have OH on the same side. But remember, the the ribose sugars, they always form the 5-membered ring. They don't form the 6-membered ring. Okay. And then we have the D-fructose. Fructose is a ketohexose. Unlike glucose, it's not a aldose. It's a ketose sugar. Okay. And the difference between glucose and fructose here is glucose, we saw that it forms a pyranose ring, a 6-membered ring. Remember that fructose forms again a 5-membered ring. The mechanism is same, but it's just that you have to remember that here the carbonyl group is at C2. So C2 will be the anomeric carbon here, not C1. And cyclization will take place between C2 and OH. This you have to remember. So fructoses form furanose rings, 5-membered rings. Glucose forms the pyranoses. Are we okay till now? Actually, as I said that fructose can also form pyranose, but it's more happy to stay. It gives it more stability. So it's more happy to stay in the furanose. So no need to go into its pyranose part. Are we okay, everyone? So what do glucose form? When glucose has to cyclize, does it form furanose or pyranose? 5-membered or a 6-membered ring in the Hayward projection?
6.
6-membered. Glucose forms 6-membered. The pyranose. What about fructose? Pyranose or furanose? 5-membered, the furanose rings. Okay. What about the aldopentoses? Aldehyde group and the pentose. 5-membered again, right? So what is 5-membered called? Furanose or pyranose? 5-membered, what is it called? You know how to remember 5-membered?
Furanose.
F for 5, F for furanose. That's how you can remember it. So you will never forget it now. F for 5, F for furanose. Okay. So this is like the same mechanism what we discussed, in the while forming the pyranose ring in the glucose. So this is the same thing. Bond between this oxygen and carbon dioxide, and this H will go then here. This becomes OH. So from this straight chain, it will form the pyranose ring. This is about the fructose. The same mechanism what we discussed for the glucose. Okay, so now moving on further about the chair conformations. About the chair conformations, I actually have a model which I always show in the classroom. Okay, which is I have made that using wire and different bonds and all that. But unfortunately, we are not in the classroom. So this I will show you once we are back to the campus, and I'll open the camera, uh, because this is something which you will not understand. You need to visualize it. You need to see exactly what I mean. Okay, so you cannot visualize this chair conformation over here. Okay, but I'll just try to explain a little bit about it, but I'll show you, and then that time just remind me, I will explain you once again. Okay, once we are once I'm back to the campus. Okay, so what is chair conformation? Okay, chair conformation is like it's one more conformation in which glucose loves to stay. So glucose can stay as an open chain. Glucose can stay as an Haworth projection, the cyclic form, and the chair conformation is basically the 3D representation of the glucose. Okay, and this is like a more comfortable position for it's more comfortable than the Haworth which we discussed. Okay, so this is the 3D representation for glucose. That's why I need to show you in a 3D model. Then only you'll be able to visualize what exactly she means. Okay. So chair conformation of your glucose gives a true picture of its 3D structure. Okay, and now your question might be why glucose wants to enjoy this chair conformation. The reason for that is because every carbon here is a tetrahedral. Every carbon here is a tetrahedral, right? And because of the tetrahedral nature of the carbon, right, pyranose sugar, they assume this chair conformation. Because as I said that, I need a model to explain that, So because if you see here, we don't want, these molecules to overlap. We don't want, the overlapping thing happens. So there's a lot of overlapping which happens in the other cyclic forms, right? So that is why glucose finds that this position is like a more comfortable than the cyclic form as well. Are we okay? That is all you need to know about the chair conformation. Okay. So as I said that this I will explain once I have a model in my hand, so you'll be able to visualize it. Okay. Why do I say that? The tetrahedral nature of the carbon. It's it's basically because of that. It favors the chair conformation. Okay. So moving on further, we'll talk about now mutarotation. Okay. What is mutarotation? Now I want everyone to pay attention. Mutarotation is basically, let's say I have alpha D glucose. There are two anomers of glucose now, right? Alpha D glucose and the beta D glucose. We discussed that, right? So let's say I have alpha D glucose, okay, and you also know the optical rotation now, guys, right? I'm expecting that you're revising the previous chapters. We discussed the optical rotation previously, right? So let's say I have an alpha D glucose, and what I did is, right, I dissolved it in water, okay, and then I tried to find its specific rotation. Remember the specific rotation, the dextro and the levo. If you remember last time in the chirality chapter, we discussed that, how it rotates the plane polarized light towards the right and towards the left, right? Yes. Okay, let's say I have pure enantiomer, I have alpha glucose, and what I did, I dissolved it in water, okay, and then I try to find out what is its specific rotation. Okay, it has been found that a pure alpha D glucose shows a rotation of plus 112. Can anyone tell me what is plus? Why did I write plus? To which side the light will be
To the
rotated?
right side.
Towards the clockwise. Very good, right? So it will be dextro, right? Plus. Okay, and then what I found is that I took the pure solution of beta D glucose, okay, and then I tried to put it in the sampler, right? And then what I saw is that it can rotate the plane polarized light this much, plus 18 point. Remember we did alpha there, the angle. So this is that same alpha, okay? Then, so this is an alpha showed the rotation of about 112.2 degree and beta showed the rotation of about 18.7 degree, okay? So when I had a sample of alpha and beta pure anomers, what I did when I dissolved them in water, okay, what I saw that slowly and slowly after some time, gradually their specific rotation changes, okay? And what happens to my surprise, the specific rotation of both alpha and beta changes and it reaches to an equilibrium value, okay, where, so this is an equilibrium value where I have the specific rotation of about plus, these are all experimental values which they have been observed, they have been done in the lab and experimentally calculated and shown. 52.7. Are we all listening carefully? Okay. Now what happened when I got 52.7, I was surprised. I was like, what is happening? I had alpha 112 degree, 112.2, and now I have 52.7. What is exactly happening? Or I had beta with 18.7, and now I have 52.7. What is happening? If you observe this, alpha has changed from 112 to, it has reduced to 52.7, isn't it? Its optical rotation changed, what, it decreased from 112 to 52.7. At the same time what I see that beta was 18.7 but it has increased from 18.7 to 52.7. I'm still not understanding what exactly is happening. But then what we calculated that, okay, let us see in this solution how much alpha and beta we have. These are all calculated values, guys, okay? So in the solution we found that there's 64% of beta I have and I just have only 36% of alpha. What is happening? So what we inferred from all this is that when you dissolve alpha and beta glucose in water, the specific rotation changes gradually and it reaches to an equilibrium value of 52.7 always, okay? And what is happening basically, more and more beta is converted. If you see here, I had beta only 18.7 and now more and more, okay, I had alpha more in number, right? So more and more alpha has been converted into the beta, because if you see here, there was a decrease in the optical rotation of alpha from 112 to 52. That means what is happening, what does glucose do when you dissolve it in water? The structure opens up like this, the ring opens up in water, okay? And for example, if you had alpha, alpha changes to the beta form. That is why I got 64% of beta in the solution and only 36% of alpha in the solution. Because initially I started with beta only 18.7 degrees, but then it increased from 18.7 to 52.7. That means we have more and more beta in the solution as compared to the alpha, right? So can I say that when I dissolve glucose in the water, okay, the most out of two anomers, the alpha and the beta, the most favorable structure, okay, for a glucose is beta. Can I say that? Yes. And you know why does this happen? This only happens because the ring opens up here, the ring opens up here, okay, and then recloses, okay. So this, it all this happens because there is the ring opens up and then allows a rotation to occur, and then it changes to the other form. So it's like, you know, it changes to, from beta it changes, from alpha it changes to the beta, right? So that is why experimentally we found 64 in 100% solution, 64% of the beta is present and only 36% of the alpha form is present. So we can say that carbohydrates like your glucose can spontaneously change between alpha and beta, as you can, as you saw here. Okay, and this happens because the hemiacetyl ring or a hemiketyl ring can open, right? So this change in the specific rotation, specific optical rotation, okay, wherein your alpha and beta were converted, okay, you know, is called as a muta rotation. So this change in the optical rotation due to the slow interconversion of the pure enantiomer of what? Converting alpha into beta, right? Gave us 64% beta and 36% alpha, which is an equilibrium mixture, and this is called as a muta rotation. Do we have any questions about it?
Doctor, could you please repeat? I don't really get it.
I know. I'm sorry because I need to finish the chapter also today. We are already lagging behind. So I'm going to explain it at the end of the chapter. Don't worry. Would that be okay?
Okay, thank you.
Okay, dear. Thank you, dear. It applies to generally to the carbohydrates. Okay. So, but here we're talking about only glucose. Okay. So generally, you know, especially when they're hemiacetals and hemiketals, they open the structure. Okay. There is a ring opening and the ring closure takes place with the hemiacetals and hemiketals. Okay. So I'm quickly recapitulating it so that people who do not get it. Okay. So what I said that whenever I have a sample of alpha and beta pure anomers, and when we dissolve it in water, the optical rotation changes. Initially, it was 112. Okay, give me a minute. Sorry about that. Okay, so what we found that the rotation changes. You know, it initially we had 112% alpha, right? And we saw that its percentage was decreased. Is it clear? Ladies, do you have any question? Okay, so I'm going to repeat it once again at the end. People who did not get it, don't worry, I'm going to repeat it once again. Okay, at the end, don't worry. Shall I move on to the next slide? Ladies. Okay. Okay, let's move on to the next slide, glycosides. Okay, so now what is glycoside formation? Okay, now please pay attention everyone, whenever a hydroxyl group of an anomeric carbon, okay, so guys, it's very, very important you revise whatever we discussed previously. So you should now know what is an anomeric carbon, right? So whenever the hydroxyl group on the anomeric carbon, let me show you the, okay, this is my anomeric carbon and this is my hydroxyl group of the anomeric carbon, right? So whenever the hydroxyl group on the anomeric carbon of a monosaccharide, so my glucopyranose or beta D glucose is my monosaccharide, right? It reacts with the OH group, hydroxyl group of another carbohydrate or a non-carbohydrate. Here it's a non-carbohydrate, but I'll show you in a while another carbohydrate, the reaction between the two carbohydrates. Look at this. For example, what is this? This is our sucrose, a disaccharide. So this is my anomeric carbon over here. Okay. So there's actually a hydroxyl group over here. So whenever the hydroxyl group on the anomeric carbon of one monosaccharide, this is my monosaccharide, reacts with the, there's an OH actually here, reacts with the OH group of another carbohydrate, okay, it leads to the glycoside formation. Are we okay? So glycosides are examples of acetals. They are not hemiketals or hemiacetals. They're acetals. Okay. And if I have to erase this, so the bond from the anomeric carbon to this OR group, this bond is called as a glycosidic bond. So the question says that how are your monosaccharides, in the disaccharide, how are your monosaccharides joined to each other? So you can say that they're joined by the glycosidic bonds. Are we okay? Is it clear to everyone? So for example, if you see in this case, I have a sucrose. So between one moiety and the other moiety, between one, this is a glucose and this is a fructose. So these two are joined by the glycosidic linkages.
So, doctor?
Yes, dear. Any questions till now? Yes, dear.
What molecule exactly joins? Is it the hydrogen or oxygen?
It is the OH. See, then water comes out to show the other reaction, the original one. Look at this here. So it is this OH and this OH which is forming a bond, right? And what happens within that, this hydrogen and one oxygen will come out as a water, right? And this is your glycosidic bond. This over here is your glycosidic bond. So it's like COC. That is your glycosidic bond. Is it clear?
Yes, doctor. Thank you.
Okay, it's in short, the water comes out, and then you have, the way I showed you, then you have O connects, because if you see here, there was an OH here. There was an OH here, there was an OH here, right? So water came out, and now they're connected with this O, these two. So this is a glycosidic bond over here. Okay. So these glycosides are very much stable. They are more stable than your monosaccharides, right? So they're more stable, right, than your monoacetals. Okay. And as I said, that they're more stable. So what about mutarotation? Is the mutarotation possible with the disaccharides? No. Whenever these glycosides are formed, there's no mutarotation because these rings, they don't open here. The way we spoke about in the mutarotation, the ring opens up here. No, these rings, they do not open here at all. Okay, so that's why you have to remember one thing, that no mutarotation in the glycosides, right? Because glycosidic bonds are very, very stable as compared to the monosaccharides. So you have to remember that it's the monosaccharides only, the glucose which we discussed, right? It's the monosaccharides only which can undergo to the mutarotation. Here, there is no mutar because this is like a locked structure. They're more stable than the monosaccharides. Are we okay? Any question till now? Are we okay, everyone? All understood? Okay. So when you have to name these glycosides, you will name them by listing the alkyl and the aryl group first. So this is the alkyl group, CH3, right? So you're going to name it as a methyl beta. Why? Because we know that the OH was actually on this side. It was beta, OH on the same side. Okay. So D, because OH was on the right hand side when when it was an acyclic structure, and gluco because it's a six-membered. It was a six-membered glucose, right? And the pyreno, right? It's pyreno. It's a six-membered cyclic ring now. And side is the word which we use for the glycoside. So pyrenose, we replace the E with the IDE. Okay. Any question till now? So you will learn that once we discuss the disaccharides, you will see that we'll talk about that. Yes, we do have the glycosidic bonds there, okay, which basically joins your two monosaccharides to form a disaccharide, for example. Any questions till now? Okay. So now we'll talk about few reactions of these monosaccharides, okay? Wait a minute. Okay, so now we'll talk about the alditol, how the alditols are formed. Okay, so we know that the monosaccharides have the carbonyl group. Okay, we know that. So as I said that you need to keep on revising everything. Okay, so foundation should be very strong. Then only you will understand. Because every time we study something new, complexity increases. So the basics should be clear. So we know that monosaccharides have the carbonyl group, right? And if it's an aldose, it has at C1. If it's a ketose, it has the carbonyl group at the C2, right? So what happens is that, for example, the carbonyl group of the monosaccharide, as you can see here, this is a monosaccharide glucose, okay? It's saying that it can be reduced to the OH group. Look at this. This was a carbonyl group. It has been reduced to the OH group, okay? And we can use many reducing agents. For example, in this case, they have used the sodium borohydride as a reducing agent here. Okay. And what is formed at the end? After finishing this reaction, we get something called as the alditol. So monosaccharides can produce the alditols through the reduction reaction. Okay. So it's basically this hydrogen has been donated by the sodium borohydride to this, and that's how we got the alditol over here. Okay, and if you see here, this alditol is also called as a polyalcohol. Okay, and we also call them as the sugar alcohols. Now, what should you know as a biochemist? As a biochemist, you should know that where is the application of this alditol or polyalcohol? Where do I apply it? Okay, so what happens? For example, there's a polyalcohol called the sorbitol. How many of you know about sorbitol? I'm sure that you would have heard this word, whenever we uh, eat a chewing gum which has less sugar, okay, or maybe if we drink a diet coke. Does anyone know what is a sorbitol? No idea. Okay, no issues. I'll explain you. Okay, sorbitol is basically it's found naturally in various berries, cherries, plums, pears, apples, seaweeds, algae, right? Now, how is it? Sorbitol is basically the polyalcohol, which we got from for example, glucose. Okay, so they basically, what happens? They are mostly marketed in the market. You'll get them as diabetic foods. Okay, you call them artificial sweeteners. People who want to reduce their weight. Okay, so what do they go for? They go for low calorie sweeteners, isn't it? Or sugar substitutes. They don't want to have table sugar, they want a sugar substitute for that. A diet coke, isn't it? Or cookies, a diet cookie, a diet or sugar free candies, right? Yes. So this is an alternative to the sugar, right? Sorbitol is an alternative to the sugar, and especially we use it for diabetic patients, right? So why do we use it for diabetic patients? Because they're totally non-caloric, or they have very, very less calories. And look at this, it's 60% as sweet as your table sugar, sucrose, right? But it will give very, very less calories. If sugar will give you 4 calories, sorbitol will give you just 2.6 calories, like half of what your sucrose is giving you. Are we okay? So we use it where we use it, as I said, that we use it to prepare the sugar-free candies, sugar-free cookies. Okay, there are some sugar-free syrups produced for the children. Okay, so this is the application of the sorbitol in the market. Okay, and very, very effective, very, very used for the diabetic patients. As I said, that they have very less calories. Now, what is the advantage? You might have seen this chewing gum in the market, right? You might have seen that this chewing gum will definitely, when they will market this product, they will definitely say that, say no to dental caries, protect your teeth. Have you ever seen this chewing gum available in the market, guys? Yes or no? Okay, so what happens basically? How do they protect our teeth? The reason for that is, okay, the reason for that is basically they do not cause, dental caries. They don't hurt our teeth, right? Because the bacteria which is present in our mouth, Streptococcus mutans, right? This bacteria, which actually causes the tooth decay, it does not use sorbitol as a source of energy. So if you eat sucrose, if you put sucrose in your tea, right? So the bacteria in the mouth, it loves to feed on that sucrose, right? And eventually produces a lot of, acid, which causes the dental caries. But the bacteria which feeds on these polyols, polyalcohols, okay, or aldetols, right? It produces no acid. So that is why it gives protection to your teeth. And the second best advantage is that, for example, if you use uh, for the diabetic patients, they do not increase blood sugar at all in diabetic patients. They have very, very, very less glycemic response. Okay, so that is why these xylitol, sorbitols, they are all used as the sugar substitutes in the diet foods, diet cold drinks, diet ice creams, right? Sugar free chewing gums. So that's what their application is. Okay, okay. Now aldonic acids. Now one more reaction which the sugar is going to give us is the formation of the glucuronic acid, oxidation to the aldonic acids. Okay, so what happens? The aldehyde group of the aldose is oxidized. Okay, and what does it form? Okay, it forms a carboxylate anion. As you can see here, this is an aldehyde group, and it has formed a carboxylate group over here. Look at this. Okay, now what is happening? So the product which is formed over here, this product, we call it the aldonic acid, D, you know, D-glucuronic acid. Okay, for example, do you know what is detoxification in the body, and which organ? As an, I'm sure that because as an anatomist, you should definitely, as a medical student, you should know that which organ in the body causes detoxification. We discussed that last semester in anatomy. Liver. Absolutely. To some extent, kidneys as well. It's mostly liver. Very good. So what is detoxification? Detoxification is, God forbid, whenever we eat anything which is, toxic, or any drug or any antibiotics, or for example, if we eat artificial sweeteners, nowadays we eat so much of food which contains a lot of artificial things, right? Artificial chemicals, artificial sweeteners, preservatives. So whenever we eat all these things, what is happening? There's a process of detoxification which takes place by in the hepatocytes of the liver, wherein the toxic substances are converted into the less toxic or more soluble. Okay. And then they are eliminated from the body. They are thrown out either through the urine or or through the bile. They're released either through the bile. Okay. And then they go out of the body. Okay. So that's like a normal detoxification process which takes place, and liver plays a very, very important role in that. Now what happens? The glucuronic acid which is formed in our body, okay, this glucuronic acid, what is it used for? It's used by our body to detoxify whenever, God forbid, there's any phenol or alcohol in the body. Okay. So what happens in liver? These compounds are converted into the glycosides of the glucuronic acid, and then that glycoside of the glucuronic acid is finally excreted in the urine. Okay. Maybe you will study that in College of Medicine, the conjugation reactions, how exactly drugs, or how exactly, the poisonous things are, are thrown out of the body. Okay. Because these conjugation products, they're very less toxic. Okay. And they're more soluble in the body, and that's why excretion becomes very much easy. Okay. So glucuronic acid is like one such conjugating agent in the body. The idea is to convert the toxic product into less toxic and more soluble, so that you can send it out of the body very easily. So that's a function of your glucuronic acid. Now what is the propofol? Propofol, for example, it's used in the general anesthesia. Okay. It's used as a drug. It comes in the form of a drug called as the Depriven, this propofol. Okay. So whenever a person has a planned surgery, okay, so in order to make that person unconscious during the surgery, we need to give an anesthesia, right? So now this propofol, for example, okay, how is it metabolized in the liver? Okay, it is metabolized only through the process of glucuronidation, wherein it is converted into the propofol glucuronide, this compound, by various enzymes in the body. So we don't want to know the enzymes, we don't want to know the procedure, just remember the names. That's it. Okay. And that is how this propofol is, metabolized and thrown out of our body, and goes out of our body with the urine. Are we okay? Or you're kind of lost? You're finding it difficult, guys? I know it is. Indeed, it is. But don't worry. I'm just trying. I'll just try to finish, and then at the end, I want to give time. So wherever you have doubts and confusions, I will. I will. I will repeat it. Don't worry at all. But at the same time, the other classes, they have all. They're going to finish lipids this week. We are not even finishing the carbohydrates yet. So that is my speed. I'm going with such a slow speed. But don't worry, I know that all this is new to all of you, and definitely it will be difficult for comprehension. But trust me, if you study every day, okay, and revision is important. And I'm here to answer all your questions. If you want me to, I will repeat 100 times. Don't worry about that. But let me finish the chapter first and then I will repeat. Don't worry at all. Don't worry at all. Okay? Because I know that all this is new to all of you, right? So it's going to take some time, once you move to lipids is little more easier than carbohydrates, so don't worry. Okay, so we'll be all okay. So glucuronidation is the process. Okay, there's a part of detoxification in the body. Okay, right. Now this is easier, like now this is easy. I'm sure that you'll find all this easier because in this you just need to remember, for example, what is the sucrose? Okay, sucrose is a disaccharide. What is it composed of? It's composed of two monosaccharides, glucose and fructose. You should know all this. Okay. And then we ask you about these linkages. The question says that for example, the question in sucrose, how is your how are two monosaccharides linked in the sucrose? How is your glucose and fructose linked? So you should memorize these linkages. You should know that it's alpha 1 T glycosidic bond. Like this is like very important in this. And obviously, what is your sucrose made up of? So all this is easier now. Is it clear? So sucrose, we know that it's a table sugar. We know that it's a disaccharide. We know that we extract it from the sugar cane, from the sugar beet, right? And look at the two monosaccharides here. This one is a glucose and this one is a fructose. Or you can also say that glucopyranose. Can I say that? Because I know that glucose always exists in the pyranose ring. And I can call this as a fructofuranose because I know that fructose exists in the form of furanose ring. Okay, so now remember, you have to memorize these linkages. That, as I said, that's very important. Okay, so if you see here, this is alpha. Why alpha? This is an alpha anomeric carbon over here, isn't it? And then this is why alpha. This is alpha 1. Okay, and then this is 2, right? So this is alpha 1 2 bond. So as I said, that this name is very important, and what is present in the sucrose, that's very important. Okay, so this is a glycosidic bond. So like this is like easier to remember. Yeah, and the water has come out. Yes, Dana, the water has, there was OH here, the water has come out, right? We saw that reaction, glycosidic reaction previously. Okay. Then we move on to the next one called as the lactose. Okay. Now, who will tell me what is lactose composed of? What are the two monosaccharides present in the lactose? It is the galactose and the glucose. So if you see here, this is the galactose. What happened to this? This is the galactose. Okay, and this is the glucose. Okay, and look at the links over here. Let me erase this. So we have beta 1,4, isn't it? Look at it. This is beta 1,4 link, glycosidic bond over here. Remember this point. What bond is present over here? Okay, so this is your anomeric OH over here, right? Anomeric carbon over here, right? And then it goes towards the 4th position in the glucose. So that's how it is the beta 1,4 glycosidic bond. Okay, and what about lactose? Okay, so galactose, okay, we know that it's present in milk. Okay, so what about human milk? Do we have lactose only in the cow's milk, or is it present in the human milk as well?
It's present in the human milk as well.
Very good, right? And this is the percentage of lactose in the human milk. And we know that how important lactose is for small infants. It's like a very, very important carbohydrate. Like they take their nutrition only in the form of lactose, right? And the enzyme which is present in them is the lactase. Okay. Now, yes, it's present in human milk. Now, what is galactosemia? Have you ever heard this about this name before, or you have heard only the lactose intolerance? No, nobody has heard this name before. Okay, galactosemia is basically a genetic defect. Never heard of this. Only lactose intolerance. Yeah, no, it's different from the lactose intolerance. It's totally different. Okay, so here in the galactosemia is what happens. Here it's a genetic disorder, okay, wherein the babies which are born, okay, they are unable to metabolize the galactose present in the milk. Gala means milk, okay, so galactose, milk sugar. So they're unable to use that milk sugar. And don't you think so? It can be life threatening, right? Because small babies, for them lactose is everything. Okay, so it becomes galactosemia is a disease which is definitely a life threatening disease. Okay, while as lactose intolerance is not, of course not. Okay, so what happens is that in galactosemia is that if they're unable to take the galactose from the milk, right, and if it remains untreated, it can definitely cause brain damage, speech problems in small children. Okay, and it's basically caused because of the absence of enzymes which we need for converting your galactose to the glucose. So galactose has to be converted into glucose. So unfortunately, they have the, it's a genetic disorder, right? So they don't have that enzyme itself which can convert galactose into the glucose. Are we okay? No, they can drink, but it's like they don't have the enzyme which can convert galactose to glucose. It's not they don't have problem with the drinking. They can drink, but inside they have a genetic problem, right? Because of that, they're unable to produce uh, the enzyme required for its conversion, for converting galactose to glucose. Okay. So lactose intolerance is different. It's totally different than a galactosemia. It's not life threatening, as I said. No, right? So you will see that like there's 60% of the people in the world, they have lactose intolerance, or as they grow towards the adulthood. Okay, so what happens, they lose the lactase activity. So lactose intolerance means that the lactase enzyme, okay, there is a reduction in the enzyme activity. So that's not life threatening. So that's perfectly okay. If you have lactose intolerance, it's okay, no problem. No, you don't need to drink milk. Okay, so adults have other sources. For example, if you're not drinking milk, you're not eating yogurt, right? So you can eat other things to improve your calcium intake. You can eat a lot of broccoli, right? Or you can, sometimes we give them tablets, lactase tablets, right? And you can add those lactase tablets in tea. Okay, so it's not life threatening. Lactose intolerance is not life threatening, but galactosemia is life threatening, because small babies cannot eat anything else other than the lactose in the milk. Any questions till now? Okay, then we have the maltose, right? Maltose is again a disaccharide. Okay, so if you hydrolyze it, okay, what will it give? Anybody? It has two molecules of This is also glucose.
Glucose.
This is also glucose. Absolutely. What is it? Remember the linkages. Alpha 1 4. This is the alpha 1 4 glycosidic bond over here. Okay. So what is this alpha 1 4 conveying it to me? Alpha 1 4 is giving me an information that the two molecules of D-glucose, these two molecules, right, are bonded between C1 carbon of one molecule and C4 carbon of the another molecule. And the C1, the alpha is the C1 is the alpha carbon over. It was the alpha glucose over here. Okay? So that was about the maltose. Then we talk about polysaccharides. What is the difference between polysaccharides and disaccharides, guys? Poly and di. What is the difference between two?
Disaccharide is made of only two monosaccharides.
Sorry, your voice is very feeble. Di is we have only.
Polysaccharides are more than 10 monomeric units,
Very good.
and they're also complex carbohydrates.
Very good, wonderful. Thank you, Rand. Absolutely, very good there. Superb. Has only 2 units, Sarah. Very good, Sarah. Abdulaziz, thank you. So polysaccharides, okay. Now, what is the main function of polysaccharide in the body? The main function of polysaccharide in our body is that they are used as food reserves. Yes, Noora, you have a question? Noora, you've raised your hand. You have a question? Okay. Maybe that was accidentally. Okay, anyways, so polysaccharides, they have two important functions. One, that they are like food reserves in the body. For example, we spoke about the last in the endocrine system, how glucose is converted into glycogen, right, to store a lot of glucose in the body. And same thing with the plants store it glucose in the form of the starch. Okay. And the second thing, polysaccharides are important in terms of, they are structurally they are important because plants, they have in the cell wall, they have cellulose. We'll talk about that. Okay, we'll first start with the starch. So again, these linkages are very important in this as well. And what are they made up of? That is also very important, guys. Okay, so plants, when they have to store the glucose, they store it in the form of the starch. Okay, now what happens if you see here, starch can be separated into amylose, remember this please, and amylopectin. Now you should know what is the difference between amylose and the amylopectin. Okay, so when I say amylose is unbranched, as you can see here. It's like a linear polymer like this. There are no branches coming out. Okay, linear polymer of a lot of glucose monomers. So you'll have glucose everywhere, but it has a linear structure. Okay, and you have almost like 1,000 to 4,000 units present in each amylose. Okay, so what you have to remember is that number one, it's linear. I mean, it's unbranched, it's linear, and the linkage is important. So how are the glucose molecules linked here? By alpha 1 for glycosidic bonds. Is it clear? So unbranched and alpha 1 for glycosidic bonds join the two monomeric, join the 2 units of the glucose in the amylose. That's one important thing. Okay. And then we have amylopectin. What is the difference between amylose and amylopectin? Amylopectin is totally different from the amylose, even though it's a component of the starch. And what is the difference? It is larger than the amylose, number one. It's a larger molecule than the amylose. It contains almost 10,000 glucose units. Here we had just 4,000 units and here we have 10,000 glucose units. And point number two, amylopectin has a branched structure. Look at this. This is a linear chain here. And then we have the branched structure. That's how they're connected with each other. So amylose was not branched. Amylopectin is branched. Okay. Now, if you see here, what are the linkages here? Okay. So amylopectin, they already have alpha 1,4 glycosidic bonds. Okay. So same like the amylose, alpha 1,4, that's already there. Okay. But after every 24 to 30 units, they also have a branch coming out. As you can see, there's one branch coming out over here. Okay. And that is called as alpha 1,6 glycosidic bond between 1 and 6. So this is an alpha carbon over here, anomeric carbon over here. So remember the linkages. So amylopectin is larger than amylose. There are more glucose units. It's a branched molecule. So the two linkages in the amylopectin are alpha 1,4 and the alpha 1,6 linkages. Are we okay? Any questions about the starch or amylose or the amylopectin? This is like an easy thing to remember, right? We move to the glycogen, right? So we all store carbohydrate in the form of glycogen, isn't it? Glycogen is like analogous to your this amylopectin. Okay, but what is the difference between amylopectin and glycogen? So glycogen is pretty much larger than the amylopectin, and there's more frequent branching in the glycogen. Like after every 10 residues, like 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. After every 10 residues, the branch comes out. Then 10 residues, the branch comes out. So it's more branched than the amylopectin. Okay. So what kind of linkages are present here? So alpha 1,4, okay, and then alpha 1,6. Isn't it? Same as an amylopectin. But as I said that, it is more branched, more complex than the amylopectin. Any questions? You don't need to know that. Then you have cellulose. Okay, what is the difference between cellulose, okay, and starch, guys? We eat both the things. We eat cellulose as well as we eat starch. But what is the difference in between starch and cellulose? Ladies, boys.
One is alpha and the other one is beta.
Cellulose is more stiff in structure. Okay. Cellulose indigestible. Very good. Absolutely. Linear. No, in our body, what is the difference between, can we digest cellulose? Okay, can we digest starch? Can we digest starch? Yes. Okay, can we digest cellulose? No. Why? Do you know why we can't digest cellulose?
Because we don't have the specific enzymes that can digest it.
Very good. Absolutely. Yes. That's what we're going to talk about here. Let's first discuss the structure of the cellulose and then move on which enzyme is absent in humans, why we cannot digest cellulose. Okay. Cellulose is basically present in plant cell walls. Okay. Plants, they have cell wall and it's present in the plant cell walls. And cellulose is so abundant, it's present everywhere on earth. It's the most abundant organic material on earth. And why do plants have cellulose? Because it gives rigidity and strength to the plant cell wall. Okay, so cellulose is like your amylose. It is linear, no branching. Okay, and contains thousands of glucose subunits. See, there's no branching over here. It contains thousands of subunits over here. Okay, here the glucose residues are joined by, this is a linkage, beta 1 for glycosidic bonds. Okay, so do you find any difference between starch, glycogen, and the cellulose? What was the linkage in starch and glycogen, guys?
Alpha.
Alpha. Very good. We have a beta, and that is a very big reason for us that we are not able to digest the cellulose. Cellulose cannot be digested by humans because we have the enzymes, like for the alpha thing, we have enzymes like alpha amylase, which is present in our saliva, in our stomach, in our gut. And we also have one more enzyme called as alpha 1,6 glucosidase. So we have enzymes to break the alpha glycosidic bonds. Okay, so for example, if we have alpha 1,6 glucosidase, we can break down the amylopectin of starch. Okay, but unfortunately we do not have the enzyme to break the beta 1 for glycosidic bonds. Humans, and there are some animals, they do not have the enzyme beta glucosidase. Instead, we have alpha 1,6 glucosidase. We don't have the enzyme beta glucosidase. So that is why we cannot digest the cellulose. But why do we eat cellulose then? Because cellulose is a very, very important constituent of the, it adds fiber. Since it's a non-digestible carbohydrate, it adds, fiber to our stools, right? It increases the bulk of the stool. It keeps the intestinal mobility, in control. It prevents the constipation. That's why we eat cellulose. Okay. Now, why cellulose is very, very strong? Okay. If you remember, I told you that starch molecules, okay, they don't give rigidity. Okay, but cellulose, they give rigidity because they have, as you can see, they're glucose molecules. Okay, it allows a lot of hydrogen bonding between the adjacent glucose molecules. So as a result, they are very, very strong. Okay, the cotton clothes which we wear, right, we get it from the cotton plant. See how strong it is. Okay, because there are a lot of hydrogen bonding between the adjacent glucose molecules in the cellulose, which is absent in the starch. Have you okay till now? Any questions? Okay. So that's why, as I said, that they have intermolecular hydrogen bonding, and this is the reason also they're insoluble in water as well. Okay, this is the same thing what we discussed. We don't have the enzyme beta glucosidases. Okay, like termites they have bacteria in their intestines. Okay, so that's why they can eat wood, because they have the enzyme beta glucosidase. And some animals like horses, they can also digest grass and hay because they have the beta glucosidase, because what we have only alpha-1,6 glucosidase. We discussed all that. Then we have the acidic polysaccharides. Okay, so acidic polysaccharides are basically, okay, a group of polysaccharides that contain either the carboxyl group or the sulfuric ester groups. So polysaccharides with the carboxyl group, okay, so they play a very, very important role in the structure and the function of our connective tissues. You know what is connective tissue, right? So whether we talk about cartilage, bones, synovial fluid, skin, tendons, blood vessels, like everywhere you will find these acidic polysaccharides. One of the very good example is the hyaluronic acid. Last time I told you that this is a very important component of connective tissue. It's present in the synovial fluid. Okay, so it's an acidic polysaccharide. And another acidic polysaccharide is the heparin. How many of you know what is heparin? What is it used for? Yes? Nobody? Not sure. Okay, let me tell you. Heparin, okay, you might have, I thought like maybe you know that it's a blood thinner, it's an anticoagulant. Okay, whenever a patient has a heart attack, okay, it's given to heart attack patients, yeah, for blood clots, absolutely, to dissolve the blood clots. Okay, given to people who have myocardial infarction or stroke, okay, or if they have pulmonary embolism in the lungs. So heparin is basically an anticoagulant. Okay, so in simple sense, you can say it's a blood thinner. Okay, so it simply prevents the clot formation. Okay, so it's sometimes given to the patients, after the surgery. Okay, and where is this heparin present? Okay, this heparin is also an acidic polysaccharide, and this is present. We have cells called as the mast cells. I think we're going to talk about them, about these mast cells in lymphatic system. Okay, so there's something called as mast cells. They're like a type of white blood cells, but they don't stain blood. These white cells, they are found, in the tissues, especially in the connective tissues, and within these cells, they have smaller granules. Okay, and within these granules, okay, they have, okay, okay, that contain your heparin. Okay, so they're usually called as the mast cells. And we're going to talk about mast cells also. Okay, we're going to talk about mast cells also in, when we talk about immune system. Okay, because mast cells, they play a very, very important role in the immune system, because they not only contain heparin, okay, within that, they also contain something called as a histamines. Okay, histamine, it's an anti-allergic thing. We usually, whenever there's an allergy in the body, okay, so we produce a lot of histamines, and that is why sometimes we eat antihistamines to subside that allergy. Okay, so in short, heparin is an anticoagulant which is present, okay, in the intracellular granules of the mast cells. Okay, and we usually find them, as I said, that in the connective tissues, okay, either in the liver, lungs, gut, right, or in the arterial wall, the blood vessel wall, you will find them there. Okay, and as I said, they also produce the histamines, and histamines, they act as a vasodilator, They dilate the blood vessels. That we'll talk about in immune system, in PHSF. But here what we need to know is that they're acidic polysaccharides and they're also sulfated. As you can see here, look at this. They're all sulfated. So if you're given this structure, so you can easily identify it by these, by the sulfate molecules present in it. So this is, you can identify, oh, this is heparin. Okay, and you should know what are they used for. Yeah, you should know the function as well. Are we okay till now? Any questions? It's okay, no problem. Then we have glycolipids and the glycoproteins. Okay, so when we say glycolipids, what is the meaning of glycolipids? Glycolipids. Glyco is the sugar part, isn't it? And lipid is a lipid, of course. Or glycoproteins. Glyco is the lipid part. I mean, glyco is the sugar part, and then proteins. Right? So these glycolipids and glycoproteins are so very important in our immune system. Okay, they're found on the cell surfaces. They act as a receptors. Okay, and more importantly, we have various blood types. Okay, in chapter of blood, we're going to talk about the various blood types. Okay, but in that you will see that if a person has a blood type A, okay, the person, we say that the person has an antigen A. Okay, we're not worried about antibodies. We'll learn that in blood chapter. You're more worried about this antigen over here. Or if a person has a blood type B, they have antigen B. If the blood type is AB, then the antigen is A and B. And if the blood type is O, we say that they have no antigen. But what exactly is this? So what happens, there are certain sugars like oligosaccharides, okay, few molecules of sugars, okay. These carbohydrates are present on our cell surface, okay, and they play a very, very important role as as a markers. For example, I know my blood group is A, for example. So this is like a marker on my cell, on my red blood cell. Okay, let's try to understand its biochemistry. Okay, this is like a structure which is present in your book. Okay, I'm sure that if you see this structure, you all will be lost. Okay, so that is why I brought a diagram here. So this can help you to understand what exactly the structure says. So what is important, you have to know, structure is not important here, but you have to know, for example, if I tell you blood group A, you should be able to know which oligosaccharide and what components are present on the blood group A. So that is why I brought this structure here, so that it becomes easier for you to understand. Are we okay? Should I start? Okay. So when I say I have blood group O, let's say I say my blood group is O, what does that mean? Okay. O is like, we call it a null group as well. Why? Because if you look at the RBC, okay, on the plasma membrane of the RBC, person who have blood group O, okay, what do they have? They have a small oligosaccharide, like they might be having glucose, they might be having galactose, they might be having N-acetyl glucosamine. Remember the amino derivatives of the glucose? They might be having galactose, and they might be also having fucose. That is it. So we call it like a type H. And this type H is present in everywhere, in O, in A, as well as B, as well as AB. It's like a basic group, which should be present in all the RBCs, on all the RBCs. But when I say that my blood group is A, for example, my blood group is A, what does that mean? That means I have this basic oligosaccharide is there. That means I have this glucose, I have this galactose, I have this N-acetyl glucosamine, I have this galactose, I have this fucose. Now look at the difference here. If I say that my blood group is A, that means the additional moiety I have, sugar I have, is the N-acetyl galactosamine, or we call it a GalNAc. So if I ask you that if a person has N-acetyl galactosamine, what will be the blood group of a person? Will it be A, B, AB, O? It will be A, isn't it? So like if you see here, this basic H antigen is present everywhere. Like look at this, the glucose, glucose, galactose, galactose, N-acetyl glucosamine. Yeah. So like this basic thing is present everywhere, but they additionally have N-acetyl galactosamine. That is why this is blood group A. And then if I say that this person has blood group B, what does that mean? That means the basic H antigen is there, glucose, galactose, N-acetyl glucosamine, galactose, but additionally they don't have N-acetyl galactosamine. Instead, they have another moiety which is called as a galactose. So with the person with the blood group B will have galactose. Now, who will tell me what will blood group, what will be the blood group of AB? If a person is with the blood group AB, what will that person have on his RBC? If a
Would they
person,
have the N-acetylic, that one, the purple one, and then also another galactose?
absolutely. What about the H antigen? H
Um,
antigen, will they have H antigen?
yes. No.
Yes.
Yes,
No,
they
no, you
will.
have to be sure.
Yes, they will.
As I said that H antigen is like these these one, two, three, four, five things are present. One, two, three, five things are present in all of them, like O, A, B, A, B, all of them. But additionally, AB will have both N-acetyl galactosamine and galactose. While as the blood group O will have neither of these two. Okay, very good question, Tashir. You don't need to know these structures. No, of course not. That's why I told you. No need to know because, I know that is very confusing. You don't need to know the structures, but yes, you need to know like what I explained you in this slide. So like if you memorize this slide, I think it's more than enough. It will make your, comprehension easier. So you need to know uh, which sugar is present in O, A, B, or A or B.
Doctor, we don't have this slide.
Yeah, I know that there, because this is an additional slide. As I said that, at the beginning of the class, I told you what we're going to do about that, right? So because I know that the other structure is very, very difficult to memorize. No need to memorize that one. Okay. So do we have any questions till now? Are we okay till now? I know this is the last slide there, Tanya. I know, dear. This is the last slide. This is like just 1 minute and we'll be done. Okay. So what is a streptomycin? Streptomycin is basically a carbohydrate antibiotic. Okay. So this is like streptomycin is called as one of the very important discoveries, in the carbohydrate chemistry. Very, very important. Now, before I will start explaining about the streptomycin, I will tell you what you should know about this. Okay. For example, if you see this structure, okay, you should know that it has streptidine, number one. You should know that it has L-streptose, the sugar, which as you can see here, this is a sugar over here. Okay. And you should also know that it has all this. So this is like the first thing which you should know about the streptomycin. Okay. Before I could explain you about the streptomycin, do you know why do we eat antibiotics? Why do we eat antibiotics? What is their function in the body? Everybody's
To
silent.
fight unwanted bacteria.
Very good, absolutely. Right. So streptomycin is like one such antibiotic, okay, and a very, very important drug, because this streptomycin has the capability of damaging the protein synthesis of the bacteria. It can damage the proteins of the bacteria. Okay. And it's simply you have to remember that it is a carbohydrate antibiotic, and you need to remember that, okay, it has these three derivatives, it has these three things present in it. That's all you need to know about this slide, right? This is a cyclohexane derivative, okay? This is like a monosaccharide, as you can see here, okay? And then this is like amino sugar. This all is an amino sugar present here, okay? So very important, okay? And especially this streptomycin is very important when it comes to, there are certain bacteria, they're very, very resistant to penicillins, whenever you have upper respiratory tract infection. There are bacteria, they're very, very resistant to the penicillins, right? So you can give them the streptomycin, okay? And also used to treat tuberculosis. Yes, basically to kill the bacteria, especially the, the bad bacteria, and they're very resistant. So you can use streptomycin to kill those. Okay, with that we finished the chapter, and I wanted to do this revision as well, but I know that you all are tired, but that's okay.